Hello everyone, welcome to the new video tutorial of Bio. In the last session we have dealt about different types of microscope. In this session we are continuing with the second part of microscopy. If you haven't subscribed to our channel, subscribe to it and enable the bell icon to get daily notifications. So without wasting time, let's get on with the video. So in the last session we have developed two types of microscope, and the first one is simple microscope and the second one is compound microscope. In this session we are dealing with the third type of microscope, that is the phase contrast microscope. So in phase contrast microscope, it was developed by the scientist Fritz Zerny. He actually got Nobel Prize for this. So that is the first point. And what happens is that the principle of phase contrast microscope is that changing phase of light, which enables the viewer or observer to observe the object or the specimen to be clearly visible. And what happens is that in the microscope, phase contrast microscope, when light passes through the object, it undergoes retardation or phase change, which differentiates the object from its background, so that the object or specimen can be viewed without staining. So that means light cells can be seen with the help of phase contrast microscope. That is a major advantage of phase contrast microscope. There is no need to fix the cells or the object or specimen, and no staining is required. We can view the living object without staining with the help of phase contrast microscope. The point to be noted is that the principle is changing phase of light, which enables the observer to observe the object, which differentiates the object from its background. So that's the main point we have to note in this one, this phase contrast microscope. Now the fourth one is interference contrast microscope. It is not that important, but sometimes the principle or a principle might be asked in the entrance exam. So I'm mentioning the interference contrast microscope here. Now what happens is that it detects small changes in refractive index of the light. So that is what happens. Changes in small continuous changes in refractive index is detected, and that makes the object visible under the microscope. And what the main purpose of the interference contrast microscope is that it is preferred for observing wet mounds of bacteria. So that is a major application of interference contrast microscope. So wet mounds of bacteria. So that is a point we have to be noted here. Wet mounds of bacteria can be observed. It is preferred to observe the wet mounds of bacteria. So that is the main application of interference contrast microscope. The fifth one is dark field illumination microscope. It's actually a modification of the phase contrast microscope. Here also the principle of light is utilized. So what the principle is that when the light strikes an object, it becomes scattered. So it makes the object luminous and visible in a dark background, So that is the main point here. So the object becomes visible in a dark background. So in a high contrast image will be formed, so that the object can be viewed clearly what all are there. So there is a dark background in which the object can be observed. The main principle here is that light, when it strikes an object, it gets scattered. So that's the main principle. Now the major application is that it's used for diagnosis of certain diseases. And another important point is that certain microorganisms which cannot be viewed through an ordinary microscope can be viewed through dark field illumination microscope. For example, syphilis spirochete can be viewed using dark field illumination microscope. So that is a major application. So the primary thing which you have to focus here is that it is used for diagnosis of certain diseases. And second one is that certain microorganisms which cannot be viewed through ordinary microscope can be viewed through dark field illumination microscope. So I hope you understood the principle and the application of dark field illumination microscope. So you should note it down because it is really important. The sixth one is UV microscope, that is the ultraviolet microscope. The major point here is that instead of ordinary light, UV light is used, and there are certain precautions are taken so that UV light does not reach our eyes. So that necessary precautions are taken, so there is no worry. So UV light is used and necessary precautions are taken so that the light from the UV light after striking the object does not reach our eyes. Usually it is viewed through a screen, or maybe a filter may be fixed in between objective lens and the ocular lens, so that the UV light coming from the object or this light source does not reach our eyes. So that is the main point. The advantage is that resolving power is increased because you have to note the equation. This one, limit of resolution is equal to 0.61 lambda by Na, and when limit of resolution decreases, resolving power increases. And for decreasing the limit of resolution, you know that numerical aperture is almost same for all microscope, but the lambda, the wavelength, can be is changed for can be changed. So lambda, wavelength of a light, should be decreased so that limit of resolution is decreased and hence resolving power is increased. So this point should be understood, then you can understand why a UV microscope have higher resolving power. So that is the important point related to UV microscope. There's just a concept or to understand the basic principle of UV microscope. So I hope you understood these microscope in a brief way. If you have any doubt or confusion, please mail bio@gmail.com. So wait till next video. Bye.